Ladies and gentlemen, I'd like to welcome you to this afternoon's policy panel and discussion. Thank you so much for coming here. I'm May Akrawi, the Consul for Science and Innovation at the British Consulate General and part of the Foreign Office's Science and Innovation Network, along with Catherine Santamaria. And you can't hear me? Okay. I just introduced myself and said that I'm the head of the science section here, part of the UK's Foreign Office Science and Innovation Network, along with Catherine Santamaria. And we've been working with the Baker Institute, Neil Lane and Kirsten now for many years on various issues of science and technology, and a subject that I know is very dear to both of our teams, and especially the interaction of science and policy. So we've done programs on stem cells, climate science, nanotechnology, and others. So we appreciate the partnership. And in fact, this policy debate is the fourth in the series of the conferences that we have done. And the first was in 2004, and we were honored that Lord Naren Patel was also there. We're very, very grateful that Lord Patel is here. He's a member of the House of Lords and a very distinguished legislator who has been involved in stem cell and fertility legislation from its inception. And we're also very grateful to have other speakers that have flown in from the UK. So you had a lot of scientists in the morning presenting on the research, and that will continue tomorrow. And we hope that a lot of research collaborations will come out of that, as they've done before. And we appreciate that we also have James Lawford Davis, Rob Buckle, Chris Mason from the UK, who'll also be joining us on the panel. And Bill Brinkley will be moderating that. So I just wanted to thank the Baker Institute, thank Neil and Kirsten, and thank the Texas-U.K Collaborative and Dennis and Lisa, especially for all the work that they've put into this. We also wanted to thank the Huffington Foundation for their sponsorship, as well as our other partners for this program. And I also wanted to acknowledge Representatives Cohen, Hardcastle, and Colcourse for taking time to come and listen today, and we hope that we are able to answer some of the questions you may have and really start a debate going. And we appreciate the other staffers and chiefs of staff who've come for other representatives' offices. So the only thing I have left to do is to introduce Dr. Bill Brinkley, who's not just a very distinguished scientist in his own right, but who's become a very good friend over the years. Bill is the Senior Vice President for Graduate Sciences and also Dean of the Graduate School of Biomedical Sciences at Baylor College of Medicine. And he's also a Distinguished Service Professor in the Department of Molecular and Cell Biology and co-director for the Keck Center for Computational and Structural Biology. If I went into all of Bill's career, we'd be here all afternoon. So I'm just going to leave you to read the rest of his biography and hand the afternoon session over to him. Thank you very much.
Well, thank you, May, and now the fun starts. I have the pleasure of moderating this small panel of experts on stem cell policy. Now, it seems to me that a major obstacle blocking us, at least in the United States, from achieving the full and remarkable potential of embryonic stem cell research and regenerative medicine and all of its promise is a maze of conflicting, controversial, and confusing policies that continue to evolve within the federal government and also at the state government levels. There seems to be two opposing groups here. Those that support enabling policies and those that support disabling policies. And although I've been dismayed at the pace of state and federal stem cell policy evolution in the United States, I admire the seemingly rational way that stem cell policy seems to flow in other countries, and especially that of our co-host here, UK. And we're going to be hearing more about how they do it. I hope I'm correct. I hope that it's not just another illusion. But since this panel is composed of 4 well-informed individuals from both sides of the pond, I believe we can look forward to considerable enlightenment on the current state of embryonic cell policy here and around. I'm not sure it's going to be a debate, because I believe all of us in this room, I'm sure most of us in this room, would like to see enabling policies make it possible for us to go forward. First, let me quickly introduce our panel members. You have in your packets the bios of some of our participants, with the exception of one who we're delighted to have here at the last minute, Miss Ellen Arnold, who was on vacation, enjoying life, and she gave it all up to come here today to participate with us, and we're delighted for that. Ellen works with a grassroots organization in Texas, the best little grassroots organization in Texas, called Tamared. It's an alliance for medical research, and it's made up of citizens, remarkable citizens, who are not, for the most part, scientists or policymakers or politicians. They're just concerned individuals, many with children with debilitating diseases that could be helped with stem cell research, that have given their time and effort to form this grassroots organization, to work with the legislature, to educate the legislature on the ways that would make it possible to try to answer the questions. They have a scientific advisory board, and I must admit that I am on that board and very honored to be on that board. But it's mostly the work that these remarkable individuals do that make it possible for what is truly, I think, a unique organization in the United States of concerned citizens. Those of you from Texas have heard, don't mess with Texas. That's a little slogan we have here about keeping it clean and as clean as possible. And don't mess with Tamared, I say, because they're tough too. So it's a great pleasure to be part of that organization. We're delighted to have her. But of course, we're also very excited to have Dr. Rob Buckle, who heads the MRC, a Medical Research Council at the UK, which is the leading funding agency for embryonic stem cell research, to speak to us this afternoon as well. And after the break, you will hear from 2 other individuals, James Lawford Davis, a lecturer in law and medicine from the Institute of Medical Genetics. Unfortunately, the 4th speaker, Dr. Paul Simmons, Professor Simmons from here in Houston, became ill. I hope it's not flu, but he's quite ill and couldn't be with us. So we're going to have instead, we're very delighted to have Dr. Chris Mason, who is a professor of regenerative medicine and bioprocessing at the University College London, and internationally known. So we're very fortunate to have these experts work with us. But let me open with a few of my own comments, if I may, with some general perspectives here in the United States. The ontogeny of embryonic stem cell policy has been contentious and anything but straightforward in the U.S. Prior to the 2001 Bush administration decision, no federal funds were used to support research on stem cells derived from either human embryos or fetal tissues. In 1996, an amendment introduced by Representative Jay Dickey and attached to a rider to the NIH appropriations bill prevents the funding of research involving human embryos, and this is designed to protect embryos or embryonic tissues derived by fertilization, parthenogenesis, cloning, or any other means from one or more human gametes or human diploid cells. However, the regulation does not apply to embryonic research funded by private monies in the United States, the kind that was used to support the early development of its stem cells by the University of Wisconsin and Johns Hopkins. NIH ruled that federal funds could be used to fund existing embryonic stem cells derivatives, but not to derive new cells. Subsequently, two presidential executive orders, one by President Bush in 2001, and that approved the use of federal funds for the first time to study existing cell lines, but prohibited the derivative of new cell lines, and another on March 11, 2009, by President Obama, issuing a new executive order that lifted the sanctions imposed by the Bush administration and legalized the use of new cell lines. Now, we're going to be hearing a lot more about this in detail this evening in the two major addresses by our distinguished individuals here. And so I won't go into any more detail. What will be the ultimate impact then of the Dickey Amendment on America's ability to fund the derivation of new embryonic stem cells in the future? How far will the new progressive vision of the Obama administration take us toward the kind of success we want, given the limitations imposed by the Dickey Amendment? So now let me move quickly to another, what I call state war on the stem cell cloning and stem cell derivation. At the level of state governments, because in the United States we have those states and given the ability to form their own laws with regard to state funding. Now you've got a terrific handout that really outlines all of this in great detail, so I refer to you that if you want to study the interesting history. But in this regard of the states and the state funding, I don't think there's a better example of a true war that's once happened here in the Lone Star State in the policies over the last 6 years. It's been a remarkable story, and therefore I'd like to, at this time, call on Ellen Arnold to come forth and begin to give us her unique perspective on the stem cell wars in Texas.
I suppose there's someone who's going to advance the...
Yeah, I think that...
Yes, it's magic.
They're coming up with it.
Great. All right. Excellent. Well, I'm happy to be here today and to have an opportunity to talk to you. I was asked to come in and talk today a little bit about the policies and politics in the state of Texas. And so I'm excited about the opportunity to talk to you about that. There's a lot has happened in Texas. I'm going to give you, and Dr. Brinkley mentioned a one-pager. Actually, it's a two-pager with the history of legislative policy and activity surrounding this issue. But I'm going to give you just a quick summary of it. You'll recall that the first embryonic stem cells were isolated in 1998, I believe. And in Texas, legislative sessions happen every other year, in odd-numbered years, and they last for 5 to 6 months. So there was no activity that I've been aware of in 1999, but in 2001 we began to see legislative activity. And this activity in the early years and continuing on surrounds efforts to criminalize embryonic stem cell research, to criminalize treatment using therapies derived from embryonic stem cell research. So, for example, if you were to travel to the U.K. for a treatment that's been developed as a result of research using embryonic stem cells, in 2003 a bill was filed that would have, when you came back to Texas with that treatment, you would have been fined, I think, up to $1 million and potentially 10 years in prison, as would your physician who oversaw that process. So in addition to that, efforts to ban funding for embryonic stem cell research and efforts to ban appropriations for public institutions that actually conduct the research. So the University of Texas Health Science Center here in Houston or UT Southwestern in Dallas. All of these have been the methods that have been utilized since 2001 relative to human embryonic stem cell research. beginning after the 2003 session, it wasn't actually January of 2004, but it was the fall of 2003, an organization was formed called Texans for Advancement of Medical Research. And shortly thereafter, about a year later, TAMR-ED, the Alliance for Medical Research, was formed. These two organizations, one of which is a C4, a lobby organization, and the other one is a C3, an educational organization, were formed after the 2003 session, when a small group of individuals, and Dr. Brinkley mentioned them, from all around the state came together because they had become aware of a bill that would have criminalized human embryonic stem cell research, and it's the one that I mentioned. It would have, had I taken my niece who has type 1 diabetes to the UK for treatment and then come back, I would have been subject to criminal charges and possible incarceration. My niece's physician as well. This was the piece of legislation that brought all that group together, and me as well, in a volunteer capacity to help fight that bill. So there are two organizations now, one again a C3 and one a 501c4, that work in Texas to preserve the legality of regenerative medical research in Texas for the purpose of curing diseases and alleviating suffering. And a huge part of what they do is, and again it's volunteers, travel the state to educate legislators, civic and religious groups about human embryonic stem cell research, and to meet with editorial boards to talk about stem cell research policy, and to work with legislators to develop legislation that is typically filed each legislative session. Beginning in 2005 was the year that we began to see the first legislation filed that was proactive or protective toward the research in Texas. Bills were filed that session to establish ethical guidelines for research that's consistent with NIH guidelines, to ban human reproductive cloning, which is the implantation of somatic cell nuclear transfer, to establish an oversight board to monitor regenerative medical research in Texas, and to fund regenerative medical research, including embryonic stem cell research. Those of us who had the privilege this morning or earlier today of hearing Lord Patel from the UK talk about the policies in the United Kingdom will appreciate that their policy has all of these things. And thus far, we have not been able to pass any of this legislation, either in one big omnibus bill or in a piecemeal fashion up to this point. Typically, what has happened is the bills have been heard in committee, but not voted out of committee, held in committee. After 2001, while bills banning research and funding for research and creating felonies were filed, a different approach or a different strategy began to be utilized and continues to be utilized today in terms of the legislative efforts. Typically, there'll be a few bills that are filed to ban research and to ban funding for research, but for the most part, the work and the efforts in those areas center around these strategies. Typically, they take the form of an amendment that would be attached to a bill that's already moving. It's either on the floor of the House or the Senate, typically. Riders on the appropriations bill. Actually, this year's rider attempt got more attention than has ever happened before, but it's actually not the first time that there's been an effort to attach an amendment or a rider. A rider's the word we use when we're talking about the budget, to the budget that would prohibit the use of state dollars. There are two types of riders. One that would prohibit the use of state dollars for any sort of embryonic stem cell research, and the other one would prohibit an appropriation to a public university or research institution if they conduct that sort of research, regardless of whether the research they do is funded with private dollars, federal dollars. So that's obviously was a very serious strategy. Another one is typically there are, and this is across the country, every session in state legislatures, legislation is filed to tighten or tweak regulations around abortion. And typically this is a type of legislation that can potentially impact embryonic stem cell research. So those take the form of changing definitions to when life begins. One example of that was to change the definition of life to being a full or complete complement of DNA, rather than, let's say, a fertilized egg. Or so there typically are efforts to change definitions in order to encompass and bring into that restriction embryonic stem cell research. I think another piece of that strategy has been to attempt to connect embryonic stem cell research to abortion. There is quite a bit of confusion, and we find when we work with policymakers that a lot of times they believe, or they've been told, that embryonic stem cells come from abortions, and that people are paid to become impregnated, to have their fetuses aborted, so that stem cells can be harvested, none of which is accurate information. So there's oftentimes that linkage is attempted. But each time, with the help of a small but growing group of legislators and very, very critical staff members, and I want to interrupt and say thank you to those legislative staffers that are here today, because we know better than anyone that you can't, and I know the legislators would agree, none of us could do our job without them. The depth of knowledge that they have on the issues that are of interest to their bosses is phenomenal, and we just absolutely could not do the work without them. So each time, and I'll tell you that in 2003, when I was assisting Tamara, Representative Rick Hardcastle was one of a very, very small group of legislators made up. It was bipartisan. There were Republicans and Democrats. There were freshman legislators. It was a teeny, teeny little group, and they were able to defeat this piece of legislation that I talked about earlier, that was carried by a leader, a political leader in the state house, in the Texas House. And had I been asked to predict our success, I would have said not a chance. But I underestimated their enthusiasm for and commitment to the issue. Now I'm going to jump ahead and talk a little bit about this last 2009, the legislation that was filed in 2009. And let me back up just a minute. I talked to you a little bit about evolution of strategies and the idea of amendments to bills. And I wanted to just mention a couple of other kinds of bills that typically are utilized to add prohibitions. One of those would be tuition revenue bond bills. And any public institution in the state of Texas knows how important the ability to utilize tuition revenue bonds to generate income for projects, for facilities, is really, really key. And it's one of the key things that institutions like the University of Texas and Texas A&M work on every legislative session. It's a way that they supplement the income that they get from their direct appropriations. And for several years, one of the strategies has been to attach language to the tuition revenue bond legislation that would prohibit the construction of any facilities or the utilization of any dollars from those revenue bonds on any sort of embryonic stem cell research. So that's another area that we've worked on for a long time. But this session was a little bit different, and Senator Steve Ogden from Bryan College Station, where Texas A&M is located, is a very, very powerful and influential legislator, probably arguably one of the most powerful senator in the Texas Senate. And this session, and he'd never really weighed in on this issue in all the years that we've been dealing with it in the Texas legislature. He's never really weighed in on the issue. And this session, he filed a bill that, if you'll, I went ahead and put the exact language in the PowerPoint to talk about the ban on state money, use of a facility that's either owned, leased, managed by a state agency for any research involving the destruction of human embryos or to support such research. So our legal interpretation of that legislation was that Direct state funding would be prohibited, as well as indirect state funding, such as in the use of facilities, the use of equipment, and the use of staff who are paid with state dollars. It struck quite a few people by surprise and got quite a bit of attention, but it pales in comparison to the attention that he got with the later act. He is the chairman of Senate Finance, and one morning we were at almost the tail end, pretty close to going to conference committee on the budget, and Senator Ogden came in one morning and laid out a rider or an amendment to the budget, and this was the rider. No funds appropriated under this act, which is the budget, can be used in conjunction with or to support any activity whatsoever, including research which involves the destruction of a human embryo. I think the thing that startled people was that there wasn't a hearing, there wasn't public input or debate. There really wasn't even debate on the committee that day. He mentioned that he had been talking to several other senators on the committee about it, and he laid it out, and they quickly voted to add it to the budget, and then some of you may be aware of that it garnered quite a bit of public attention. The implications, I think, are really important to be aware of, and that is that had that rider gone on the budget, any research conducted with private or federal or state dollars, even research used on the approved President Bush's approved stem cell lines would no longer be able to be conducted within any state facility. Those of you who attended the session this morning for policymakers, Kirsten talked about how there were so many public institutions in Texas that conduct embryonic stem cell research that she could not fit the names of all of them on the page. And so it would have had an impact on every entity, every public institution virtually that does research. There's also been a big concern about the impact to the ability of these institutions to attract and retain researchers. And then lastly, the belief that an act such as this would have broader impact on other fields of medicine. In the end, Senator Ogden never sought a hearing on SB 1695. With the help of the majority of the members of the conference committee, which are the group of people in the Senate and the House that come together to work out the differences on the budget, the majority of the members on that conference committee were not in favor of that rider. And he pulled the rider down, and then he filed a new bill that asks for reporting. He wanted public institutions to begin to report to the state on the type of research, because one of the comments he made was, and I find that it's very common among legislators and not a criticism at all, but he was not aware that any embryonic stem cell research was being conducted in public institutions in Texas. Not any. Now, you remember I just said that the page wouldn't hold them all. And he didn't realize, and I believe him, that what his rider would have done would have had such an impact in Texas. In the end, he added to a bill that Senator Watson had filed, and Senator Watson's senior staff is here today. He joined with Senator Watson, a key Democrat in the Senate, and they were able to attach to Representative Branch's HB 51 from Dallas a study. So what's going to happen over the next 18 months or so is that a study is going to be conducted to determine the best way to gather data on what kinds of research is being done in Texas, and it's with the goal of increasing collaboration. I'm probably running out of time. Am I out of time? Just about. So I'm just going to quickly go through this last little bit here. I want to talk just a little bit about why it's happening in Texas. I think that it's very easy to demonize those that are on the other side of our position, and I think that's a misguided way to approach it, and I think it's, in my personal opinion, it has more to do with a genuine lack of understanding about what the research is, confusion about where stem cells come from, that we only want to be able to use fertilized eggs that currently are now being discarded from IVF clinics. I think that this has a tremendous amount to do with the struggle that we've had to establish ethical guidelines in Texas. And that is that over time, the composition of legislative districts in Texas has changed, and they are much less evenly divided between the parties. And so the battles in in elections tend to be within the primary. So if I'm a Republican and I'm running for re-election, I'm not really going to have a battle with a Democrat in the general election. My battle is going to be with another Republican. And in those circumstances, oftentimes the most conservative of the two will be the one to prevail. And if you kind of take that the next 2 or 3 steps down the road, that also tends to be, those voters are more conservative, and they're the base that's elected you, and there is a feeling that you need to represent those perspectives. And I think it's had quite a bit to do with how policy has developed or failed to develop. I think there's truly a very understandable tension for public institutions because they receive their appropriations, virtually all of their funding from the state. And it's very difficult for an institution who is at the Capitol every day working to make sure that they get their appropriation to talk about a controversial issue like embryonic stem cell research. I think there's also a reluctance to discuss the economic impact of all of these ongoing battles, because these are the people that are also trying to recruit from the UK, Australia, New Zealand, Singapore, scientists to do this research. And so we don't want to talk a lot about what the environment is like here, because we really want these folks to come and work with us. And then a belief by many legislators that this research is not happening in Texas anyway. And then there's the let some other state do it attitude, or some other country. I am going to stop right here because I think I've run out of time. The last 2 slides that I had just gave a little bit more detail about the interim study. The one piece of legislation that did pass this session was an interim study that I referred to earlier, that would take a look at how to collect data on what the institutions are doing. And Senator Ogden asked that one of the technologies that have a data collection process set up is in the area of biomedical research, specifically embryonic stem cell research. So over the next 15 months, they'll be looking at how to collect the data in such a way that it doesn't put our research scientists at a competitive disadvantage, issues about what's legal to disclose and what's not. And then they will come back in January with a bill that recommends exactly what type of data collection system ought to be utilized and the kinds of information. All of the major universities in the state will be included on the committee that's put together, and so they'll all be represented and be able to give input. And so that is where we are right now in Texas politics.
Thank you, Ellen. You certainly have fired us up here, and I'd like to call on Dr. Buckle now to give us a more sane and clear, well-stated policy. Welcome to Houston.
Okay, thank you very much. Just let me open up my slides here.
Put it on for you.
Okay, while I'm waiting for the slides to come up, firstly I'm delighted to be here, and thank you for the invitation to come and talk to you all. By way of introduction, I look after a certain section within the MRC. I look after neuroscience and mental health research, and also regenerative medicine. And I'm primarily on the research funding and strategy side, but I also have a role in helping provide the secretariat for the governance committee that oversees human embryonic stem cell research in the UK. And I will come on to that in a second. So the Medical Research Council is the equivalent effectively of the National Institute of Health in the US. We're the primary government-funded organisation that funds medical research, and we have an annual budget of about $1 billion per annum. Bit louder? Sorry. Let's put that up. Is that better? You can hear me now? Okay. How's that? Better? Okay, so I'm still waiting for
They're working on it.
the slides to come up. But what I would like to do is provide my talk into three parts. Essentially, just to give you an overview of the current regulations and the current picture in the UK. Then how we govern human embryonic stem cell research in the UK by way of oversight and guidelines. And lastly, just to talk about the regulatory structure in terms of developing any therapies through to the clinic. The UK regulatory environment for this area is really characterised by primary legislation that provides the framework under which human embryonic stem cell research can be done, coupled to a licensing arrangement by which committees review proposals to undertake research involving human embryonic stem cells and human embryos, and that these are licensed. And this flexibility means that the very fast-moving science that exists in this area does not continually have to be taken back to Parliament for primary legislation to be enacted. And this system has worked fairly well. We've reviewed public opinion in the UK on this matter several times over the last decade, and it has been sort of widely, the majority of the population in the UK is widely supportive of this arrangement. So we're still having some problems with the slides here. The sort of background to the regulation that exists in the UK has really developed over the last 15 years. And I won't go into the history of that. That's Lord Patel will talk to you all over dinner about the story of how that's developed. But what I am going to concentrate is telling you exactly where we are right now, and also at the end some of the issues that are still sort of causing debate in the UK, and recognising that this is a fast-moving field and we have to be flexible in how we approach the area. The UK's regulation is primarily, it provides a framework which has clear lines and demarcation, but is considered to be permissive to the research area. And as I said, as long as the research is strongly justified in terms of the ultimate aims of the research and a rationale as to why human embryos or embryonic stem cell lines need to be used for this research, then that can be permitted under licence. So as I go through the talk, I will try not to lapse into terminology that you won't understand, but if I do, then please do alert me to the facts and I'll try and clarify things. I mean, the other point to say about the state of the science in the UK is it's broadly considered in the UK that to push medical research in this area forward, we have to progress equally well on all sorts of stem cells, whether embryonic, fetal or adult. And most of the top research centres in the UK that work in this area actually utilise all types of stem cells because the information that gained from the research. Okay. Okay, so there are three primary bits of legislation that I need to refer to, and we'll just briefly walk you through. There's the Human Fertilization and Embryology Act, which was first brought through the House of Parliament, House of Lords in the UK in 1990, before human stem cell research was perceived as possible, and then amended in 2001 to reflect the state of the science, and has recently gone through Parliament again, and so we now have a 2008 Act, and it's that I will refer to. So I will also talk about the Human Tissue Act of 2004, which is the act that governs the storage of human tissue and cell lines. The Human Fertilization and Embryology Act relates really to gametes and embryos. And finally, in terms of legislation, to recognise that in the UK we operate under the umbrella of the European Union, so there is legislation at a European level that we need to be responsive to. And there is a European Cells and Tissue Directive that also governs at a European level the storage and transfer of human cells and tissues between countries. And what is important to know is that as an act of law in the UK, it is prohibited to implant any embryo into a woman unless it has been fertilized. So that's an important thing. That's punishable by a jail sentence. So reproductive cloning at any level is not permitted in the UK. And obviously that's not a situation that you yet have in the US. So when we talk about what is possible with human embryonic stem cell research, please bear that in mind. Okay, can you give me the next bullet point? Okay, so I'll give you the key principles of the Human Fertilization and Embryology Act, which, as I mentioned, governs the generation of use of human embryonic stem cells. And the real issue here is that the legislation reflects the fact that the human embryo has a special moral status. Can I have the next, please? So the regulatory authority empowered to issue licenses for such research is the Human Fertilization and Embryology Authority. Next slide, please. And it's a criminal offence to carry out any research in this area without a license from the Human Fertilization and Embryology Authority. Next, please. So the Act allows the generation and use of embryos for research. So researchers can either use supernumerary embryos that are spare from in vitro fertilization programs, or in exceptional cases actually generate those embryos for research in its own sense. Next slide, please. Embryos can be generated by, as I said, in vitro fertilization, or in the UK by somatic cell nuclear transfer, and that is the scientific term for therapeutic cloning. And this is the process by which, in principle, you can take an egg, you can remove the nucleus from the egg, and you can insert a nucleus from another cell type within that egg to trigger the embryonic process. And that is the technology that's been used for animal cloning. Now, somatic cell nuclear transfer may be something that researchers wish to use in the future, and the UK regulation does permit that to be done. But as I said, next slide, please. This can also be done under the new regulation using animal eggs. So there is an issue of whether you can get enough human eggs to undertake this research, and scientists have argued that they do need the capacity to be able to use, for example, either cow eggs or rabbit eggs to be able to do the research at the very fundamental level on this technology. Next slide, please. But of critical importance is there's a 14-day limit for research on human embryos or any of the embryos that might be created through the admixed or interspecies approach. The 14-day limit is important because that is the point at which the embryonic structure first starts to appear with the primitive streak, and before which it's considered there is no possibility of neural development in the human embryo. Next slide, please. I should also add that human embryonic stem cells are derived from embryos typically at the 100 to 150 cell stage. And that's when they are no sort of larger than the tip of a pinhead. And it's at that point, after 5 days of development, that human embryonic stem cell lines are typically derived. So the legislation limits research to such that is, in the terms of the Act, necessary and desirable to study either fertility treatment and contraception, congenital diseases and miscarriages, and the development of treatments for serious diseases. And any applications to use embryonic research is judged against these criteria by the HFE Act. Next slide, please. So I mentioned in terms of human embryonic stem cells first being described in 1998, it was recognised that the Act needed to be amended in 2001 to take account of where the science was. And it was recognised that the stem cells, when taken from an embryo, were no longer the subject of regulation by the Human Fertilization and Embryology Authority. Next slide, please. So it's considered there was a need to put in place an oversight system for human embryonic stem cell research. Next slide, please. And alongside that, the need for a national stem cell bank, which would ensure that these very precious resources were optimally used and there wasn't an unnecessary proliferation of stem cell derivation. Next slide, please. So it was made a condition of any licence from the HFEA that any human embryonic stem cell line derived within the UK must be deposited in the National Stem Cell Bank. Next slide, please. And accordingly, this meant that the Medical Research Council was asked to help set up a UK stem cell bank, and I'll come back to this in a few slides' time, and an independent steering committee, which is currently chaired by Lord Patel, who will be speaking to you over dinner. And that committee has responsibility not only for the UK stem cell bank, but the use of all human embryonic stem cell lines in the UK. Next slide, please. Very briefly, I mentioned the Human Tissue Act. Next slide, please. Which is essentially there to govern the removal, storage and use of human material, and applies to all human cells and tissue. Next slide, please. With the exception of hair and nails of any living person, or the gametes and embryos that are regulated under the HFE Act. Next slide, please. And there's another authority, the Human Tissue Authority, that looks after that aspect of the regulation. Next slide, please. And I mentioned the EU Tissue and Cells Directive, which was implemented in 2007. Next slide, please. Next slide, please. And it's the primary aim to ensure the quality and safety of tissue and cells used for human application at consistent standards across the European Union. And the UK obviously has to adhere to that. Next slide, please. Next slide. So turning to the UK Stem Cell Bank, just to give you a sense of how this is working in practice. This was established in 2002 as the world's first stem cell bank. Next slide. It was decided that it needed to have a neutral location. It shouldn't be associated with a research institute, which could provide a conflict of interest. And in itself is not allowed to undertake any research on the human embryonic stem cell lines, apart from technologies to do with storing those lines and characterising those lines. Next slide, please. It's funded by the Medical Research Council and the Biotechnology and Biological Sciences Research Council. Next slide, please. And currently, there are 70 human embryonic stem cell lines that have met the criteria for approval, that are registered with the bank. 28 of these are from overseas, including the US, other European countries, India and Australia. Next slide, please. And 16 of these lines are currently being distributed as quality-controlled, robust cell lines for research. It actually takes nearly a year to work up these human embryonic stem cell lines when provided by a laboratory to a point where they can be reliably distributed. And this is important because we obviously feel that the role of the bank here is to ensure that research being undertaken with these lines is of the highest standard, using reproducibly qualified stem cell lines that are consistent from one lab to another. Researchers in the UK can access lines directly from the labs that derive them. They don't necessarily have to go through the bank. But obviously the ambition here is to make sure that the bank has the quality kite mark, if you like, that means that researchers will wish to use the stem cell lines from the bank, and researchers will wish to deposit their lines within the bank for distribution. Next slide, please. Next slide, please. So the GMP facilities are also part of the bank, which means that in principle stem cell lines can be provided of the quality that could ultimately be used for human transplantation. There are no such lines existing in the UK at the moment. And the other point is that the bank is set up as a UK national resource where the stem cell lines are freely available to UK researchers. And they are also freely available, aside from shipping costs and some costs in workup, to anyone overseas. So in principle, anyone in the US or Texas can apply to the UK Stem Cell Bank to use those lines. But as I'll come on to, that application is reviewed by a committee to check that there are certain criteria in place. Next slide, please. And yeah, just to say that the UK Stem Cell Bank is part of a large international coalition trying to improve international standards and make sure there's harmonisation to allow transfer of lines across the world. Next slide, please. So in terms of oversight, an independent steering committee was set up. As I mentioned, it was chaired by Lord Patel, who's been in that role for nearly 7 years now. Has a very broad membership involving scientists, ethicists, lay members. The Department of Health and regulatory bodies are also involved as observers, and MRC provides the secretariat. And as I mentioned previously, the role of that committee is to review all applications to use human embryonic stem cell lines in the UK. Next slide, please. Next slide. So below that, is a local management committee for running the bank. Next slide, please. And there has been, until very recently, a committee of users and clinical scientists who have been there to highlight issues about how the regulatory processes are impacting on their research, so that we can get the best balance between satisfying the research community and the demands for regulation and oversight that the general public would wish to see in place. Next slide, please. So I mentioned the constitution of the steering committee, which, as I said, is totally independent. Next slide, please. The steering committee has a code of practice for how the stem cell bank is run and how human stem cell lines are used in the UK, and this is available on the MRC and UK Stem Cell Bank website, but I should mention it's currently under revision to take account of recent advances in the science. Next slide, please. The committee reviews all applications to, next slide, use human embryonic stem cell lines in the UK. Next slide. Import and export human embryonic stem cell lines, and deposit and access stem cell lines in the bank. And the next couple of slides, next slide please. Next slide please. I'll just mention how this works in practice so you can understand the sort of scrutiny that applications go through. So any researcher wishing to use a human embryonic stem cell line has to submit an application to the UK. This is on a project by project basis and on a stem cell line basis. So if the project was to be modified to use another line in due course, they would have to reapply to the committee. Next slide, please. The form identifies the principal investigator in the institution, the identity, source and provenance of the stem cell lines in question. Next slide, please. The nature of the project, so we can understand the aims and objectives, but there is no effort to peer review the project as would be done by grant funding committee. Next slide, please. We ask, of course, whether the project in question has been peer reviewed, and if it hasn't, we will put it to a different level of scrutiny. Next slide, please. We check whether the application is to use the stem cells for research use in a laboratory setting or for clinical use. And as I've already mentioned, we haven't got to the point of using these stem cell lines in a clinical setting as yet, although that's probably likely to happen in the next couple of years. Next slide, please. It's important to understand whether the research involves the use of animals, and again, we would have extra scrutiny if there was that possibility. Next slide, please. And again, whether it involved the use of aggregating embryos and mixing with animal systems as well. Next slide, please. But the committee doesn't have any role in ethical approval, which is left to local ethics committees in the UK, or reviewing the use of animals, which is also reviewed under a separate process through the Home Office in the UK. Next slide, please. So very briefly, if you want to deposit human embryonic stem cell lines in the UK Stem Cell Bank, next slide, please. The committee reviews whether there are essential licences in place, the approvals are correct, and that the appropriate consents and accreditations are there. So in particular, has the patient donor information been correct? Is it fully understood how these lines might be used in the future? And has that all been done under fully informed consenting process? Next slide, please. We want to check that the derivation is in accordance with our code of practice. Next slide. Next slide, please. The sort of technical data to make sure that all of these cells that are in the bank are actual bona fide cells that will be of use to the research community. Next slide, please. We, of course, need to check on who owns the line and what intellectual property arrangements are there to check there's nothing inhibitory there. Next slide, please. And whether there are any conflicts that might impact upon the line being deposited or for embargoing its release, which some derivers sometimes put in, especially for lines that have been generated overseas and maybe wish to be put into the UK bank. Next slide, please. For lines derived overseas, we realise that the consenting process cannot be absolutely identical to the UK, but the committee is charged to checking that there is equivalence of standards in terms of fully informed consent and patient information, donor information. Next slide, please. Okay, I think I've said that. Next slide, please. So very briefly, in terms of the current issues with the Code of Practice, we're reviewing the Code of Practice, have been consulting. As of always, there's perceived bureaucracy in this process, and we do our best to explain to the researchers why this system is in place and why it actually protects their research in the long run. But we recognise we need to make it as efficient as possible so that research isn't inhibited. Next slide, please. There's an issue that is currently coming into play about how human embryonic stem cell lines might be commercialised by companies. And if you recall, a requirement in the UK is that all lines derived in the UK are put in the UK Stem Cell Bank and made available for researchers. And companies, as they're moving towards clinical development, are questioning whether this might be in conflict with the ability to commercialise these lines and actually build therapies. And that's something that we're working out a way through at the moment. Next slide, please. That really, as I said, boils down to the requirement for exclusivity in how these stem cell lines might be used from a company's perspective versus the original altruistic donation of the donor. Next slide, please. Next slide, please. So induced pluripotent stem cells have been discovered in the last few years and have really excited the field. These do not fall under the legislative aspects that I've talked about. They do not involve the destruction of embryos. Nevertheless, it's considered that there are ethical issues associated with the use of these lines, and the committee is evaluating really what they are and what the guidance should be. But I mean, in general terms, current procedures for ethics review, especially around donation of the tissue, are covered by the regulatory guidelines already in place. Next slide, please. And as I mentioned, the Code of Practice is due to end at 2009. Now, I should probably stop there in interest of time. I could go on to talk about, I've got a few slides on the governance of regulation towards clinical development of therapies, but as I say, in the interest of time, it might
we will have a chance to talk some more on the Q&A session if you'd like to, but if you wish, go ahead.
No, that's fine. I think it's a good break point. So I apologise for the disruption in presenting that, but I hope that was informative.
Okay, I'd like if you and Ellen would come forward, and Arcatel, I believe if you'd join us, we'd like that at the front table here, and we can have questions and discussion from the audience, and I hope there will be. You must surely have thought of some things that we can have questions from the audience. Yes.
Actually, this is for Ellen. I was actually curious because you didn't give too much details about the data collection that they're trying to study, I suppose. So is your opinion that this is something that's good for the state of Texas to do this collection? Is this going to be something that's then in the end they're going to go, wow, we're doing all this research, wait, we don't want to do all this research, this is bad? I mean, what direction, or is it kind of neutral right now? Is it maybe a good education and maybe they'll be more supportive of it when they have a better idea of what's going on?
So the question was, and you're asking my opinion, but the question was, relative to the intent of this study about data collection on various technologies. I think I have to believe that the intent of the legislation is very good, and that the original intent of the legislation is to facilitate collaboration among research entities within Texas, and how that plays out and what it looks like when it's all said and done, I'm not certain. The fact that all of the public institutions are going to be included on the committee that makes those decisions, I think, is really important. And one of the reasons that I wanted to be sure to talk about it today to this audience was to make sure that you're aware that these decisions are getting made in the next 15 to 16 months. Is there a scenario in which knowledge about what institutions are doing human embryonic stem cell research might have an impact on a particular institution? I suppose that's not outside the realm of possibility. But I think that the original intent of Senator Watson was certainly collaboration and helping the research to move forward rapidly in Texas. Doesn't it worry you? Well, I have to believe that. That it's for good intentions. And I will say this too. I will say that over the 6 years that we've worked on these issues, that more and more legislators have become educated on regenerative medical research. And I think that if there were an attempt to somehow misuse that information, or I think there are plenty of legislators that brought to their attention would, I think, attempt to address that.
I think there's a question right here.
California set up the $3 billion fund. Do you have any information how that's working out, and any other states planning to do the same thing?
I don't have a whole lot of specific information about other states. There have been more than a dozen states that are funding embryonic stem cell research with state dollars. I know, my understanding is in California, and Dr. Brinkley may know better, there have been any number of lawsuits that have sort of slowed that process down. It's my understanding that they've kind of gotten on the other side of that now.
Hopefully, yes. I think there's some promise that it's moving forward now with the universities.
But it's taken quite a bit longer, I think, to get started than anyone anticipated that it would due to lawsuits.
Okay, yes, question right here. I'm sorry, I can't see well enough to identify you.
Horst, Dr. Buckle, right? Question about when you said, when we're talking about the stem cell bank, and you said it's open to UK and overseas researchers. How does that work? Do they come to you? How does that, give me an example.
Okay, there's the application forms are on the internet, so that basically any researcher interested in using a line picks the appropriate form, fills that out, and it's submitted to the committee. And they can be from a researcher in the UK just wishing to use a line, or, for example, a researcher in the US who may want to access a line from the bank. And there will be an appropriate form for that. And then that is reviewed on a 3-monthly basis. So applications come in 4 times a year. The committee sits and reviews these, and basically can either approve or decline the application depending on the information provided, or generally if there's a decline we'll ask for more information because it's usually clarity that's required. Of course, the agreement requires that if any research is to be undertaken in an overseas country using these lines, the committee will take the view that the research is bona fide research, and the line they wish to use has been ethically sourced, i.e. it's in the UK Stem Cell Bank's registry. But it's incumbent on the researcher overseas to adhere to whatever national or state guidelines might pertain where that person's doing the research.
And a follow-up. You talked about, and I'm sorry I didn't get this down in my notes, you talked about one of the committees that kind of oversees but doesn't do any of the ethics oversight because it's done more locally, or I wasn't sure.
Yeah, so sorry for the confusion. That's the same committee. So there's one oversight committee that looks at all applications to undertake human embryonic stem cell research in the UK. Now these are by and large applications to use cell lines, and if there is ethics committee approval, which is done at a local basis generally, either through the university or the hospital that may be undertaking the research, then that is where the ethical review of the project is done. What the committee will require is that there's proof that ethical review has been undertaken, and that all of the paperwork is in place for that. But it's not part of the jurisdiction of the committee to undertake any further ethical review of the science.
My last follow-up. Laura Patel, close your ears. If you were starting over again, would you do anything differently? I kind of traced in your remarks, it was kind of evolutionary in ways, and you started in 2002 and you kind of added and layered up and kind of where you are today. If you were starting over, would you change anything?
Personally, I don't think so. I think we've achieved a balance which has kept the majority of the researchers happy and the majority of the general public happy. We have surveyed, as I mentioned, public opinion on this and the legislation several times, most recently at the end of last year, and that was one of the biggest opinion surveys undertaken, and there's a report referenced in some of the paperwork that you may have, and that showed very strong support for the legislation and the way that it's working in the UK. And as I say, we have on the other side of the coin regular interaction with the research community to try and refine the process and make sure that it's fit for purpose for wherever the research is. And we do have to revisit this, and we will continue to have to revisit this because the research changes. And 3 years ago, no one envisaged induced pluripotent stem cells could exist. And I don't think we yet understand exactly how they will be used and what ethical problems might arise from their use.
Would Lord Patel change anything?
No, I don't think so. I think it is important to know that the regulation related to human embryo research was there before stem cell research came, and that every time the legislation has been changed or added to address the science development, as long as the bounds of that science research has been what would be general population would consider ethical to do. So, for instance, we instituted a ban on reproductive cloning 7 years ago now, because it was quite clear that the public will not consent to that. And therefore we had to put a block to that. Not that anybody was trying to do it, but it could put a block to it. Anybody trying to come to UK and using it to do reproductive cloning. So I think the principle of monitoring the science and keeping the public opinion on board as to where the science is going, and if the public was satisfied that where the science was going was acceptable and that there was a regulation in place to have that confidence on the side of the public, then the legislators legislated accordingly. So it's been a partnership between scientists, public opinion, and legislators, with controls in each part that hitherto has worked. And the legislators have been happy to go along that line. At the end of the day, if the research does not require to use embryos, human embryos, then the regulatory authority will not allow that research. There will be no license granted if that research can be done any other way.
I'd like to follow up, if I could, on a similar question, Lord Patel, since you're speaking. Do these bank cells apply to all entities, private, federal, and university the same, or do you have any kind of dichotomy like we have?
No, any embryonic stem cells created in the United Kingdom has to be deposited in the bank, no matter who buy.
Yes.
I'm sure there must be other banks also worldwide. Can you name a few countries you also have a bank? And if you collaborate with these countries, and also is it a goal to get all the, I don't know what's the latest number, 300 cell lines? Is it your goal to get all of them?
Well, I think Dr. Buckle will tell you about other banks, but before he does, if you ask me, my ambition would be to create the world's biggest bank and the only bank.
So how far are you on that road?
Pretty close.
We brag in Texas, too.
Yeah, but this is not just bragging.
You'd make a good Texan, Lord Patel.
Are y'all the only bank in the world?
No, I mean, in answer to that question, no. There are other stem cell banks operating worldwide. You sometimes have to look a little behind the website to understand exactly how they're operating, but there are a number. I mean, there's one operating out of Harvard. There's one in Singapore. There's some stem cell lines in Australia being distributed through one lab, but essentially a bank. And another number of other countries. Spain has set up in Europe a stem cell bank as well. Firstly, there is an international activity trying to bring all of the banks together to share common guidelines and establish for those countries that may wish to create a new stem cell bank, what are the minimum expectations for this to work. Secondly, I mean, in some ways, as Lord Patel indicated, we would argue to some of the people that we talk to, why set up your own bank at high cost if you can produce the lines and get them distributed and banked through the UK Stem Cell Bank, for example, or one of the other two global banks around. As long as the standards of regulation are in place and everyone has total transparency on how they're ethically sourced and any restrictions that might be put in place on their use, then that should operate at a global level. So that is the ambition. And I think it's important, obviously, that the number of cell lines out there doesn't proliferate out of control with no purpose, because the reality is that actually probably only 20 or 30 lines are typically used by 90% of the researchers in the world at the moment.
In that time, do you know something about that cell line that Geron uses for the first FDA-approved clinical trial? Is that correct, here in the States?
Yeah, it was one of the so-called presidential lines, Bush lines, that was created. Interestingly enough, there was quite a lot of debate or sort of discussion in the community as to whether such a line, which was derived using animal products in a lab situation which wasn't considered clean for clinical purposes, could then be used for a clinical study. But Geron have convinced the FDA that they could rederive this line under what are called GMP, good manufacturing practice conditions, and that is the line that's being taken forward. But I think year on year, new lines will be described that will be made to a higher and higher standard. And certainly in the UK, we expect to have 25 lines in the next 2 years that will be produced in total freedom of any animal product.
I think it's important, frivolity apart, it's important that the bank is just not a bank where somebody goes along and says, I got cell lines, can I put it in a box somewhere? The bank, it has to be maintained at a very high quality level, that these lines are kept on high and worked on to improve the quality. That's why I think, as Dr. Buckle said in his presentation, it takes a year for the scientists working in the bank to improve the quality of these lines to a point where the researchers can, if they access these lines from the bank, can be assured that the quality will be guaranteed. And therefore their research has a better chance of success, as speaking as a non-scientist.
Thank you. Joe Brown, president of TAMR, has a question.
Yes, I do, and TAMR being what is known as a C4, which is the more political of the two organizations. I'm thinking I'm among primarily scientists, but I am thinking in terms of politics, Lord can tell. And the way that the United States is established, we have the federal government, we have the state governments, which are very big, and especially the state of Texas, on something called states' rights. And the question really becomes, part of it is the states individually, which has something to do with the ethical approach you were talking about, but the states individually can legislate darn near anything they want for what is acceptable within that state. For instance, the state of Michigan, back at the time of Dolly, decided that they were going to outlaw any form of cloning, period. During the last election, by the way, that was a constitutional amendment which was removed in the last election. Do you see this as being a major problem in the United States, being able to actually compete in the world of science when we have got just a complete either no regulations, over-regulation, wonderful regulation, and all of this is in the mind of the beholder. And how does a country that is established with our type of governmental structure do the sort of thing that the UK has done, which is, do you have the equivalent of a state? I've never heard of the equivalent of a state in counties, yes, but is there the equivalent of a state and a state government in UK? I know you've got Ireland. I know you've got Scotland.
Well, we have, coming from Scotland, we have a different country. It's called Scotland. And I think we have another country south of us called England. But it's the UK law that governs certain issues that are not devolved to the parliaments of Scotland or the assemblies of Wales or Northern Ireland. And part of it is this related to the subject that we're talking about, embryo research. It is not devolved to the government.
Which means you have a more controllable.
But yes, there are certain other issues too, like finance and defense isn't either. So there are UK laws that govern all the other constitution. But I wouldn't presume, speaking personally, I wouldn't presume to tell you, people of America, what they should and should not have. All I would say is that you do have, undoubtedly, not only in Texas but other parts of the United States also, world quality scientists. And it's a dialogue between the legislators and the scientists and the citizens that should decide what laws should be formulated that might best benefit citizens of the United States and hopefully also citizens of the world if you find a cure for serious diseases.
If I may, I just want to clear up one thing. I was not trying to say
No, I understand that.
Anything should be dictated by anybody else. I'm just saying, it appears to me we have a different structure within which we have to work.
I believe that's clear. I'm going to have to move on. We have some other questions over here yet.
I wanted to ask about one particular point with respect to production of cell lines. You mentioned that probably most of your cell lines are coming from excess embryos from IVF, but it also says that you can have cell lines derived from other sources. And I wonder how that's regulated.
Well, it's all regulated by the Human Fertilization and Embryology Authority. So any research on a human embryo, whether it's the creation of an embryo or the manipulation of a surplus embryo from an IVF program, has to basically, the researcher has to request a license from the HFE to undertake that work. So that is reviewed by that authority.
But that doesn't tell me how the review is organized, what are the considerations involved, and that sort of thing. In other words, the production of embryos has impact on at least one other person, namely the woman who produces the eggs. So, can you go out and hire people to produce eggs?
No. It's a Human Fertilization and Embryology Authority. If I was a researcher want to carry out research on human embryo, I have to apply for a license to do that research to the Human Fertilization and Embryology Authority. It will then decide whether the license should be granted or not on the basis of, one, whether that research is necessary. Two, can that research be done without the use of human embryos? Three, can that research be done on embryos that might be available that are not used for the purposes of treating infertility? And fourth, even if all those were satisfied, is the research ethical? Primarily, is the research ethical? And if all these things are not possible, then the license may be granted for the creation of embryo for that research. Now, let me give you numbers so that might put it in perspective what we're talking about. Since 1991, about 124 licenses have been granted by the authority. Majority of those were to improve fertility treatments. About 11 licenses have been granted since 2001, 2 involving process called Sept to create lines for disease lines that carry genetic information for research, 2 for mitochondrial diseases out of the 15, and 1 for studying the maturation of eggs. But in totality, only 124 licenses have been granted since 1991.
Yes, the lady right here.
That's top of my memory, but I think I'm right.
We're here on a personal basis, as probably some of you are. My daughter's had stem cells 3 times for her multiple sclerosis. And what I'm hearing is that when y'all talk about stem cells, you are limiting it to the embryonic derived stem cells. That's what I've heard for this discussion. It's been limited to the embryonic. My question is this: if it were broadened to include, for example, she had received adult stem cells from umbilical cord, which carries no ethical issues with it. I mean, it seems like, my question is, why is there not more focus on basically the waste product, which has done her a world of good in her disease, instead of in the more politically confrontational, and maybe because I'm not a...
No, we're focusing on human embryonic, and my colleague did, partly because we have strict regulations that control human embryonic stem cell research. We recognize that adult stem cells do not have the same ethical issues, and we promote all kinds of stem cell research. We make the point in every debate we've had in the legislature that we wish to promote all kinds of stem cell research that could possibly lead to treatments that could benefit humanity. And in terms of cord blood stem cells, we are creating right now banks of cord blood stem cells, probably in excess of our needs, but nonetheless, we are creating banks of cord blood stem cells. There's a private-public partnership doing that. But the bank can curate all kinds of stem cells. But the problem is, as the scientists will tell you, about curation of other stem cell lines is not the same as embryonic stem cell lines. So do not have the view that we are only focusing on embryonic stem cells.
If I can just briefly add to that, I mean, one of the issues is in terms of regulatory purposes, the use of adult stem cells is broadly covered by standard regulations. So bone marrow transplants have been happening for leukemia treatments, etc., for a number of years, and the ethics approval and the standards for that apply across to the sort of treatments you're describing. As a funding organisation, we actually invest more on adult stem cell research than we do on embryonic stem cell research. And in fact, we're funding a number of early phase clinical studies using adult stem cell therapies. Currently, there are none in human embryonic stem cell therapy, although we anticipate they will come online as the science and the quality of the lines matures. So it's very important that there is a mixed economy and everything is done in parallel.
And I would say absolutely in Texas that our position is the same. Adult stem cell research is widely funded and certainly by entities that I represent very, very broadly supported. But what we know now is that its application is somewhat limited, and that we believe that the potential for therapies and treatments as a result of embryonic stem cell research is broader. And again, because it has the ethical considerations, then we feel really strongly that we need to establish ethical guidelines and restrictions.
Can I follow up? Is the public aware of this? When you talk about stem cells, you have a knee-jerk reaction. They immediately think embryonic. When, like for her disease, for example, the adult stem cells do a lot of good for multiple sclerosis. And I don't think the public is aware of that, and that's my final comment or question.
Well, I would say certainly in Texas that we have found that to be the case. And I think, people are busy working as hard as they can, making a living, raising their kids, taking care of their parents as they age. And I think that we've found in the 6 or 7 years that we've worked on this issue in Texas that certainly public citizens often aren't really aware of the potential for the different forms of stem cell research.
It's the lady right here in the white jacket.
Yes, hi. I'm Francesca Vitelli from Hispanics. I had a question to take us back to a little bit about the plans for the GMP laboratory and the facility. You mentioned currently there are no human embryonic stem cell based trials going on in the UK, and actually Geron's in the US has been currently placed on hold. So if you can tell us a little bit about what the plans are for your facility, and also if you can make some speculations about timeline and perhaps first indications that are most likely to result.
Okay, yeah.
And the lines will have to be, you mentioned re-derived without, or maybe derived ex novo, and create new lines that need to be recharacterized. So how is that transition going to occur?
The question is, how soon will you have clinical application?
Yes, and what are the plans for the GMP facility currently that you have in place?
Yes, there are 6 GMP facilities in the UK that are able to, at the point of taking the embryo and creating the human embryonic stem cell line, are in place. That whole process could be done under GMP conditions. We have recently funded 3 centres with MRC funding, public funds, to create the lines that I was talking about earlier. So we would hope in the next couple of years to have a number of lines that will be freely available through the bank for research purposes. Currently, there's the Geron study underway, which is using a re-derived line, and I think in the next couple of years there will be other trials put in place that may use similarly re-derived stem cell lines. And one area that is very likely to be doing clinical studies in the next 18 months is for retinal repair in age-related macular degeneration, which is a major cause of blindness, is currently incurable, and there's a good indication that human embryonic stem cell therapy may deliver some treatment benefit. There are a number of other disorders where human embryonic stem cell lines are being characterized with the potential to use, and they're numerous neurological disorders, bone and cartilage disorders, cardiovascular disorders. Some of these disorders can also be approached with using adult stem cells as well. So I think probably in the next 5 years we will see a number of trials using human embryonic stem cells. And in terms of induced pluripotent stem cells, which everyone hopes will deliver some real benefit in the area, I think certainly for the next several years we're not going to be in a position to use those clinically, because there are a number of problems in terms of the safety of how they're used that need to be ironed out. But obviously everyone is hopeful that in due course they will be a good source of studies as well.
Okay, we're going to come back after the break and have more public discussion and presentation. So I want to thank our panel here in the first session very much.
Good.